This is part 2 of the lecture on Protists. We ended with a slide that showed how the protozoans and algae are divided into groups, or how they are classified. Now we're going to look at those individual groups. Remember, they're called super groups. So the first one that we're going to look at is called the Excavata, or sometimes called the Excavates. Many of them are asymmetrical, meaning that there is no way that you can divide them in half into equal halves. Like with human beings, for example, we could be divided into a left and a right half that are symmetrical to each other, that look like each other. But the Excavata are single-celled. They have a feeding groove. And the reason they're called Excavata is their feeding groove is excavated from one side. So it's only on one side of the body. There's a variety of protists or protozoans in the Excavata. There are the heterotrophic predators, there are photosynthetic species, and then there are parasites. The subgroups are the diplomonads, the Parameciums, and the euglenozoans. Now, we have already seen the euglenozoans in the form of a euglena. We saw that, in fact, I can go back. This was an illustration here, picture C. So this is an example of a protozoan that's a member of the excavates. Okay, and we also have the subgroups diplomonads and Parameciums. For the diplomonads, one of the most common, most well-known is called Giardia lamblia. This slide is a Giardia lamblia right here. That's a slide of a Giardia lamblia. You can tell that it has flagella. They do have the remnants of mitochondria, but they are called mitosomes. That they're essentially non-functional. They exist in anaerobic environments and use processes like glycolysis. Remember, glycolysis was the first step of cellular respiration, and it is also the first step in fermentation. But it does not require oxygen. So diplomonads are anaerobic, and they have 2 nuclei. They also move by using flagella. Giardia is an intestinal parasite that can make you very, very sick. And then we have an example from the subgroup Euglenozoa. We already saw one example, which is just simply called a euglena, but here is a euglenozoa, which is also a parasite. It's a parasite called Trypanosoma brucei. See if trypanosoma is on here anywhere. No, so I'll write it. And this parasite, Trypanosoma, causes the disease African sleeping sickness. And like malaria, it's transmitted by an insect. But instead of the mosquito, it's the tsetse fly. So you can see the life cycle of the tsetse fly and how the Trypanosoma brucei is transmitted to humans. So just remember that the Trypanosoma brucei is an example of a euglenozoa, and it causes African sleeping sickness, and it's transmitted by the tsetse fly. Then we have the Alveolates. So now this is a new supergroup. So we did the supergroup Excavata. Now we're doing the supergroup Alveolates. And the chromalveolates are divided into two subgroups, the alveolates and the straminopiles. So that's down here at the bottom. Now, some of the chromalveolates are going to be unicellular, some are going to be multicellular. There are some types of algae in the chromalveolates. So let's look at the alveolates first. I remember them because of the little air sacs in our lungs called alveoli. Little tiny air sacs in our lungs. That's supposed to be an E. The alveoli in our lungs are air sacs. So it says the alveolates are named for the presence of an alveolus, which is an air sac that is beneath the plasma membrane. We don't know exactly what it does, but they have it, so that's where they get their name. The alveolates consist of the dinoflagellates, the apicomplexans, and the ciliates. Now, we've already seen a ciliate. The ciliate that we saw was the paramecium that contains cilia all around the outside of its cell. Right here, this is a ciliate because it contains cilia, which helps it to move. The apicomplexans consist of definitely some parasitic examples, and the dinoflagellates are going to be the ones that cause, if they grow, if they're kind of have an overgrowth. When you have an overgrowth of the population of dinoflagellates in the ocean, you have something called red tide, and that can make the shellfish toxic to eat. So here's a picture of a dinoflagellate, and like its name suggests, it contains, I think it's 3 flagella, but I know it contains more than one. It says dinoflagellates can be photosynthetic, heterotrophic, or mixotrophic, so all different types of metabolism. Many are encased in plates of cellulose. Cellulose is also found in plant cell walls. And then 2 flagella fit into the grooves between the cellulose plates, and then another flagella extends longitudinally. So here is 1, 2, 3. So I was right, it's 3 flagella. I thought so. And they spin. That's how they move, they spin. Dinoflagellates. So these are in the group alveolates, subgroup alveolates, supergroup chromalveolates. There are protists that contribute to bioluminescence, and that is a bioluminescence that appears in the ocean when it is dark, kind of like fireflies. They live in freshwater and marine habitats. They're part of the organisms that form plankton, so they're a food source. And not all, but some generate light, and that's called bioluminescence. So some dinoflagellates generate bioluminescence. Then we have the ciliates, which includes the paramecium. And that's the only one you need to know. Ciliates ingest their food particles through an oral groove. And of course, they move by cilia. And they're surrounded by a structure called a pellicle, which is an extra protection that doesn't inhibit them from moving. This is an image of the paramecium again. It does have a primitive mouth called an oral groove for ingesting food, and it has an anal pore for excreting food. So here is the oral groove, and here is the anal pore. So much more complex than some of the protozoans, much more complex than bacteria. And then the movement is through the cilia. It is a freshwater protist, so it needs to be able to get rid of excess water. It takes on water because it's the water outside of the paramecium that the paramecium lives in is hypotonic to the paramecium. So the paramecium is always filling full of water. So it has to have a way to get rid of all that excess water. And it uses the contractile vacuole to do that. And that vacuole is spelled incorrectly. Here it is spelled correctly. Okay? And then we have sexual reproduction in paramecium. This shows you the life cycle of a paramecium. I don't always stop to go over the life cycles, but I will here. So let's start with number 1. First of all, it's important to know, and you may have noticed this in the original illustration that I showed you, but the paramecium has a macronucleus and a micronucleus. And there are 2 different mating types. One is shown here with a blue micronucleus, and the other one has a red micronucleus. So what happens is 2 paramecia line up and form a cytoplasmic bridge. In step 2, the micronucleus undergoes meiosis, and so 4 haploid micronuclei are generated, but 3 are going to disintegrate, which leaves you with the original and then the new one. So I'm sorry, that doesn't happen. I'm sorry. I didn't read this before I said that, so forgive me. I'm not going to re-record for that mistake, but I will tell you, they after meiosis, 3 disintegrate, so you're left with 1. So there's 1 left, 1 micronucleus left in each paramecium. And then that one divides by mitosis. So in step 3, you end up with 2 micronuclei. And then 2 of them switch. They switch nuclei with each other in step 4. And then the micronuclei fuse, forming a diploid micronucleus. So now, before, this micronucleus was haploid, which we can put an N for haploid. But this micronucleus is diploid. It contains a copy of the genes from one of each mating type. So this is actually representing a fused micronucleus. That's 2 of the micronuclei that have fused. And then the micronucleus undergoes 3 rounds of mitosis, and that produces 8 micronuclei. The original macronucleus disintegrates, and 4 of the micronuclei become macronuclei. And since there are 4, that paramecium divides into 4 separate daughter cells. So this is the result of sexual reproduction in the paramecium. Okay, so those were the alveolates, and now we will talk about the straminopiles. The straminopiles are the second subgroup of the chromalveolates, and they're going to include some algae and some heterotrophic protozoans. They have a hairy flagellum, and many also have an additional flagellum that lacks hair. Members range in size from single-celled diatoms to multicellular kelp, which is a type of algae.